is the sponsor for this series of lectures. I'm glad that it's back up and running. There will be a new technology for us to explore and make mistakes in. So while we're at it, while I'm speaking, it's not that I'm a hesitant speaker, it's that I'm also checking people in while the introduction is happening. So I mentioned that FOAM is the sponsor for this series of lectures. Our next one will be on January 15. Jamie Carlino, I almost messed up that last name, will be the speaker, and the title for that talk is Rats. Now that's not a title that is about our vermin friends. The subtitle is Raptors Are the Solution. So it's about toxins that humans put in the environment and a way out. I think I indicated that questions will be welcomed after Mark is finished talking. So let me introduce our speaker. Mark Wilson will be talking tonight on the natural history of microorganisms. Mark grew up in Maryland. Apparently he was lucky to have a lot of nature around him, learn to draw and identify the basics of biology. He's been an HSU professor since 1998, and during that time has had lots of adventures, like helping to lead a National Science Foundation microbial observatory at Lassen Volcanic National Park, which I would really like to know more about. So once again, the title is The Natural History of Microorganisms. And as you know, that is the secret life of the Arcata marsh. So this goes right to the heart of what we're all about. Please welcome Mark Wilson.
Thank you very much, Elliot.
Okay.
This is my dogs are going to go off as well. So, when I was younger, I was always interested in natural history, birds and wildflowers and mushrooms, and I always felt most comfortable in nature, and it was always what I was primarily drawn to. And in graduate school, I got interested in bacteria, and I was studying nitrogen fixation. So 80% of our atmosphere is nitrogen, but most organisms can't use it, and in fact, nitrogen is a limiting element in most ecosystems. But there's bacteria that can fix nitrogen. And so we were studying nitrogen fixing bacteria in this lab, and it was in an organism that was isolated and in pure culture. And I would ask questions like, well, how important is this organism in nature? And then how important are free-living nitrogen fixers relative to the endosymbionts that have symbioses with plants? And how important is, how much nitrogen is fixed? These kinds of questions about sort of basic natural functions. And nobody had any idea what the answers to them were. I mean, we knew a little bit about microbiology because we had pure cultures and we would use very, very reductionist techniques. And I ended up being really unsatisfied with that. And I left my graduate program because I was so unsatisfied with it. I wanted to know about nature. And after a couple of years of working for different groups, I was lucky enough to find an advisor at Cornell University who was interested in the same kinds of questions and who was a wonderful mentor to me and taught me a lot about epistemology, and he taught me a lot about perseverance, and we sort of got to explore nature together. And I was also fortunate there to meet my late wife, who had a similar path to me where she was studying bacteria and got interested in bacteria and nature. And we were able to spend most of our careers together as professors, both as professors at Humboldt State with our offices next door to each other, teaching classes together and doing research together. So what I want to talk to you about tonight is this sort of worldview of a microbial ecologist, an environmental microbiologist, how somebody who is interested in nature, but particularly interested in bacteria in nature, how they sort of view the world and what they think about and what we've found and this kind of thing. And so my vision is that you're a birder, or you're a botanist. You're somebody who likes nature, who likes going for walks at the marsh, for example, and doesn't know a lot about bacteria. I think everybody is aware that bacteria are more than causative agents of disease. They're way more than germs. And so I want to just sort of introduce that world to you so that when you go on a walk, you can think about bacteria from the perspective of a microbial ecologist. So let's see if we can do that. Okay. I'm trying to share my screen here, and hopefully this works. And this is, they all seem super self-evident. Bacteria are small. Bacteria are really, really numerous. Bacteria are everywhere, and bacteria are very diverse. But I want to explore those dimensions of those very obvious things, because it turns out that there's all kinds of interesting phenomena that let us understand nature in different ways. So this is a picture of some bacterial cells. These are E. coli cells on the head of a pin. So this is an electron micrograph, and it gives you a feel for the scale of bacteria. And we'll talk about the scale of bacteria as we go along. Okay, so bacteria are too small to see, generally speaking, but we can still see the effects of bacteria all the time. So this is a picture of Grand Prismatic Spring at Yellowstone National Park in the Midway Geyser Basin. And it's beautiful, and I hope you guys have all been there to check it out. It has these gorgeous colors, and it's very, very charismatic. And what we're looking at are a bunch of different photosynthetic bacteria that live in the hot spring, and the water comes up at a certain temperature. It cools as you get farther and farther away from the center of the spring. And so you get different pigments that are produced by the photosynthetic bacteria. And there's other bacteria that we can see as well. So this is a picture, not as pretty maybe, but it's from Cooper Gulch Park in Eureka. And you often see this kind of iron film. It looks like oil is floating on the surface. Here's another picture of similar situation. So on the left-hand side, that's a little trough that's dug next to the path in Cooper Gulch Park. And you can see all of these iron oxides in there. And these it looks like an oil sheen that's on top, but it's not actually oil. Anything that forms a certain thickness layer on top of water will refract water in that way. On the right, it especially looks like oil. The picture on the right is from the frog pond at the Arcata Marsh, that little area by the log pond. And one time I was walking by and I saw this sheen on there. And so these are also bacteria. And so we can see the bacteria sometimes, even though we can't see the individual cells, we can see the effects of the bacteria. And similarly, this is Arcata, the Humboldt Bay at low tide. And we're used to looking at the murmurations of the shorebirds and the beauty of the shorebirds. But when it gets to be low tide, we get to experience bacteria that we can't see, but we can smell. And so there's a certain smell to the low tide mud, and that's hydrogen sulfide. And the hydrogen sulfide is produced by bacteria that are in the mud. And so when the mud is covered with water, the oxygen that they're using to breathe runs out. They start using sulfur to breathe. And as a byproduct of that, they make hydrogen sulfide. And this hydrogen sulfide gives the smell to the mud at low tide in Humboldt Bay. And we sometimes see little mats, like this is on the left there, and this is algae, but the purple part of that is bacteria. And so there are times when we can see bacteria, we can experience bacteria on these kinds of scales. And then on the right-hand side is the experiment that most people have used to study microbial ecology. So this is a Winogradski column, and people do this in grade schools and high schools, and you take marine mud or mud from the Humboldt Bay we use all the time, and it works great, and you put it into a glass cylinder, and sometimes you put some paper in there or something to act as a very sort of slowly available carbon source. And what happens is you get these different layers with different colors. And so this red layer here is the same kind of bacteria as we see on the left in the purple. And this is called a Winogradski column. And the idea behind it is that by setting up the mud in the glass cylinder, you create gradients. And so there's high concentrations of hydrogen sulfide in part of that and low in other parts. There's high concentrations of oxygen in part of that and low in other parts. And so each of the different environmental conditions selects for some kind of bacterium, and hence you get a big bloom of that bacterium so that you can see it. And this sort of leads to one of the most canonical statements in environmental microbiology, in the natural history of microorganisms. And that is this saying, everything is everywhere, but the environment selects. So when we look at the concentration of cells in natural environments, the bacterial cells in natural environments, they're really astoundingly high. So it's typical for something like the bay water to have 10 million cells per milliliter. A milliliter is about a teaspoon of liquid. And so, you know, when you have 1 million cells, or if you're talking about soil, it might be 100 times that many, you often have a wide variety of organisms that have been deposited there, but aren't necessarily growing rapidly or thriving or anything like that. And what we see is that you can take environmental samples and then select for something, and you'll get those organisms growing regardless of where you took your sample. This isn't always true, but as an example of that, I've done a lot of work in hot springs, and we isolated organisms from Lassen Volcanic National Park hot springs, and then Hawaii Volcanoes National Park hot springs. And what I eventually came to realize is that I can go out into my backyard and take a gram of soil and get those exact same organisms. If you put them at pH 2 and 50 degrees C, then that's what's going to come up and grow. And it's really true for almost every gram of soil that I've tested, and I've tested stuff in Florida and throughout California and Maryland and a lot of different kinds of places. And it seems to be ubiquitous that these hot spring organisms, which can only grow at very low pH and high temperature, are nonetheless widely distributed everywhere. So this is sort of an interesting maxim of microbiology, and it has to do with them being so small. So when we think about some of the consequences of being small, okay. When we think of some of the consequences of being small, one of the consequences is that cells can move around very, very rapidly. So on the right-hand side of this is a picture taken from space of a dust storm that's leaving Africa at the bottom, traveling across the Atlantic and hitting South America. And so there's bacteria all over those dust particles, and there's bacteria in clouds, and there's bacteria on birds that are migrating, and there's bacteria are moving all over the globe all the time. And so when we think about biogeography, we usually think about dispersal limitations of plants or animals. And with bacteria, dispersal is perhaps not an issue. Maybe it is when we talk about some environments, but it isn't for lots of environments. And that's a consequence of being very, very small. Another thing that's unique to bacteria is that bacteria are really good at persisting in environments where they can't grow. And so some bacteria can make spores. And there's a picture of a spore, an electron micrograph of a spore on the top left there. And these spores, they have very highly protected DNA inside them, and it's covered with these coats. You can boil spores, you can treat them with acid, you can hit them with UV light, you can do an amazing variety of things to spores, and the DNA inside survives just fine. And then when you take that spore and you put it into conditions that are appropriate for growth of the organism, then it turns into vegetative cells and starts doubling very rapidly. So on the right-hand side there is a bee encased in amber. And this is an example of how long spores can last. People have taken and sterilized the surface of the amber and then sliced into it, and there's bacterial spores in the stomachs of some of the insects that are entombed in the amber. And these are things that are tens of thousands or hundreds of thousands of years old, and the spores germinate as soon as they put them into a liquid growth medium. And there's lots of other kinds of ways that people have tested this, things like very old, like liquid inclusions in very old salt crystals and things like this. Recently, there's been this really fascinating study done by Japanese scientists where they're drilling very deep into the ocean floor, the sediments underneath the ocean. And this summer, they pulled up cores from 2.5 kilometers below the ocean floor. So the temperature's getting very hot there. There's a lot of pressure on those organisms. There's not many nutrients. It's a tough place to persist, but they found lots of microbes in these sediments that they were pulling up. And one of the interesting things is not all of the microbes were spore formers. So it wasn't that they formed these dormant states, it's that the cells were able to live for a very long time, probably not growing and doubling, but taking in enough energy to at least replace damaged molecules and persist over very, very long periods of time. So this everything is everywhere and the environment selects is a really sort of important idea to have in mind when you think about bacteria. Okay, so another bacterium that we can, am I muted? No. Another bacterium that we can experience by smell is Streptomyces. And Streptomyces is one of my favorite organisms. What we can see here on the left-hand side is the forest behind campus, and there's these nutrient agar plates that are laid out on a log. So this is part of an experiment that I do with our introductory biology students. We go out into the woods and we get bacteria, and then the students study those bacteria throughout the course of the semester, including things like antibiotic production and competition between bacteria. And what we do is we just open the plates up and let them sit there for a little bit, and cells will land from the air onto the plates. There's nutrients that are in those plates, so we put a cover on it and we put it into an incubator, and then the bacteria grow and form colonies. And one of the things that I like about Streptomyces is that the colonies are beautiful. So the idea is a single cell lands on the plate, and then that cell becomes 2, 4, 8, 16, 32, etc. And when you have billions of cells, you can see the colony with your naked eye, and so you can tell that a single cell landed there. So at the top of this figure are some colonies of Streptomyces, and going from top to bottom are how the colonies change over time. So if you look over towards the right, these are some of the kinds of things that you see when you look at Streptomyces colonies, and they're really beautiful. They're not just little white orbs or something like this, and so they're fun. But the other thing about this is that Streptomyces make this compound called geosmin, and geosmin is what we smell when we take a walk in the woods and it hasn't rained for a while and it starts to rain. Or you can smell this in your yard a lot of times too. There's that smell as the rain starts hitting the earth and these compounds start getting aerosolized. And most people love that smell. I love that smell. And that is something which is produced by Streptomyces. So interestingly, this is also the same molecule that causes that earthy taste in beets. And it's one of my favorite parts of beets. Okay, so we can experience bacteria. We can see bacteria sometimes in mats or when they're photosynthetic. They're not all completely invisible that way. And interestingly enough, I want to start talking about cells, but there are some bacteria that you can actually see with the naked eye. So this is a bacterium called Thio Margarita Namibiensis, which is a pretty beautiful name. And that means sulfur pearls of Namibia. And you can see these bacterial cells with your naked eye, And they're sort of interesting. So most of the cytoplasm, most of the proteins and DNA and stuff like this is actually in the outside here. And this is a big vacuole that's in the middle of the cell. And those little white, pearly looking things are sulfur granules, elemental sulfur. And so Thio, Margarita, Namibiensis can be seen with the naked eye. You don't even need a microscopic microscope in order to see these cells. And I mean, they're tiny. They're right at the limits of resolution for eyes. And maybe I couldn't see them without glasses, but nonetheless, you can see them. And this picture here is showing, excuse me, a Thio Margarita cell laid on top of a fruit fly. And so you can see about how big it is. It gives you a feel for how big it is. So this is a very large bacterium. There are some other very large bacteria. This one is Apulopicium fischelsonii. And this is a bacterium that's much larger even than Thio Margarita. It's up to 600 micrometers in length. Okay, so let's talk about size for a little bit because we need to talk about that to talk about bacteria and being small and what it means. So we know what a meter is. A meter is about 3 feet in length. And hopefully everybody has used rulers that look something like this that have centimeters marked off and then centimeters that are divided into millimeters. So the blue rectangle is a centimeter. That's 1/100th of a meter. The red rectangle is a millimeter. That's 1/1000th of a meter. Okay, so 1/1000th of a meter. If we can think about the size of that red square relative to a meter, then we can think about the size of bacteria. Because the red square is 1/1000th of a meter, most bacteria are 1/1000th of that red square. So that red square is 1 millimeter. Most bacteria are about 1 micrometer. So if you were laying bacteria end to end, you could lay 1,000 of them across the width of that red square. You could lay 10,000 of them across the length of that blue rectangle. And if we think about this in 3 dimensions, if you have a cubic centimeter, then you have 10,000 bacteria by 10,000 bacteria by 10,000 bacteria. So in something about the size of a sugar cube, you get about 1 trillion bacteria, 10 to the 12th bacteria. Okay, so they're small, but we can at least sort of envision what we're talking about because we know what a millimeter is relative to a meter, and so we can sort of envision what a micrometer is relative to a millimeter. Okay, so now we're gonna look at some small bacteria, and this is Prochlorococcus, and this is a pretty small bacterium. So the scale bar here is 1/10 of a micrometer. So this bacterium is not quite 1 micrometer, it's probably 1/2 a micrometer in length. And remember that a micrometer is 1/1000th of a millimeter. So this gives you a feel for the size of it. So the other thing that I want to talk about here is pictures. So the different pictures are taken using different kinds of microscopy. And this is one of the big methods that we use to study bacteria because they're smaller than what we can see with the human eye. And so on the bottom right, we're using light microscopy. And light microscopy is about 1,000 times better than our eyes. Our eyes can see things that are about 0.2 millimeters apart, and light microscopy can see things that are about 0.2 micrometers apart. So we can see bacterial cells pretty well with light microscopy. Here's an individual Prochlorococcus cell, but we can't see a lot of structure. So we use electron microscopes, which use magnets to focus electrons rather than using lenses to focus light, and we can get much higher resolution. So what we see when we look at Prochlorococcus with that is all of these structures that are inside the cells, and those are membranes. And so the old view of bacteria was that bacteria don't have internal membranes. We know now that bacteria have tons of internal membranes all the time. And these membranes are used for photosynthesis because Prochlorococcus is a photosynthetic organism. Okay, so that's a transmission electron micrograph. We can see inside of cells with that. And then the other micrograph here is a scanning electron micrograph, and we can see the elder surface of cells with that. So we can see external structures like flagella, and we can get a feel for how the organism looks in 3 dimensions. Okay, so microscopy and our knowledge of bacteria has been around since the 1600s, and the first person to see bacteria was using this thing that doesn't look much like a microscope on the right-hand side, but it's one of the earliest microscopes. And these microscopes were used by people who dealt in fabrics to try to quantify thread counts. And so Leeuwenhoek, who was the first person to observe bacteria and make observations of bacteria, used his microscopes to study linen and to look at, fiber counts in linen. But one day he looked at some dirty water and he saw all these organisms swimming around, and then he did things like he scraped off his teeth and looked at them through his microscope, and he was able to see lots of cells. He was able to get about 200x magnification with this. So he could see things that were, a couple of micrometers in size. Okay, so Prochlorococcus, that's the little cell that we were just looking at, is pretty small. It's smaller than a micrometer, and it is probably the most abundant photosynthetic organism on Earth. So Prochlorococcus is responsible for about 50% of the photosynthesis in the ocean. So this is a really important organism from an ecosystem perspective. And in terms of the oxygen in our atmosphere, probably about 5% of it comes from Prochlorococcus. It might be the most abundant genus on Earth. And very interestingly, it was first described in 1988. So there's been some pop science articles about Prochlorococcus, and says sharing is paused. Are you guys still seeing my, I'll reshare. Are you all seeing a shared screen still? No, you're not. Okay.
It just came back.
Stop?
Oh, now we're back to you.
Okay. I'm going to just get back to that screen then. Okay. Here it is. Okay. Okay, so because Prochlorococcus is so important in the environment, there are pop science stories about it. And this is a figure from one of those pop science stories about it. And this is something that happens all the time in microbial ecology and in microbiology. The numbers that we talk about are immense. And so people try to come up with analogies so that we can sort of understand how to experience them. And so in this particular article, they said that the mass of Prochlorococcus cells in the ocean is equivalent to 220 million Volkswagen Beetles, which I thought was a pretty interesting way to try to describe how much mass of them there is. And then the other thing that's on here is it says there's about 100,000 cells per milliliter of ocean water. So I wanted to talk about that a little bit, how we know how many cells there are. And so there's a couple of different techniques that we use. And one of them is just direct microscopic counting. So we use this device that's over on the left-hand side here. And this is a specialized microscope slide where you can put a little sample of water into a groove that's on the slide. And then there's a cover slip that's a specific distance away from the bottom of the slide. And so the water sort of wicks in there, and we know exactly what depth of water we are looking at. And then when you look through the microscope, there's these fine lines that are etched into the bottom of the slide. And these are precisely machined, so we know exactly what volume of cells we're looking at. And for example, we can count the number of cells that are present in a square like this. We know exactly what volume we're looking at, so then we can figure out what the concentration of cells per milliliter or per liter is, something like that. So this is one way that we figure out how many cells are present in a sample. And then another way we figure out how many cells are present in a sample is we take our environmental sample and we dilute it. So this is a series of 1 to 10 dilutions. And then you take a known volume of your dilutions and you spread it onto a nutrient agar plate. And once you get dilute enough, you get separate individual cells. They grow and divide and form colonies. You can count the number of colonies and then back calculate what the concentration must have been in your original sample. So this leads to another sort of interesting feature of environmental microbiology, which is called the Great Plate Count Anomaly. And the Great Plate Count Anomaly is the difference between counts you get with direct microscopic counting and with plate counting. And what we see is that oftentimes for every 1,000 cells you see under a microscope, you only get one that grows on your nutrient agar plate. Sometimes it's even worse than that. Sometimes a little better than that. But when people, for example, first started studying marine microbiology, for every 1,000,000 cells or so that they could see, they would only get one colony growing on a plate. And so the original interpretation of this was that the ocean was a vast graveyard of dead cells that couldn't grow and do anything. And ultimately, people did a set of experiments where they added things like radioisotopically labeled sugars and amino acids. And then what they saw is that those things got taken up by these cells. So clearly the cells were living and they were able to do things metabolically. They just didn't feel like forming colonies on our plates in the laboratory, or we weren't giving them something that they wanted, or they need to be forming associations with other bacteria or something like this. So there's issues with all of these approaches. And oftentimes what we do is we use multiple approaches and see where these different independent approaches converge. Another problem with this is that not all bacteria form colonies. So this is from some work that we were doing at Lassen Volcanic National Park. And we knew these bacteria were present in our samples, both because other people had studied them in similar environments and because we were doing gene surveys and we could tell that these organisms were there, but we couldn't get them until a graduate student, Francine Arroyo, she went on to do a PhD at Cornell and she's doing a postdoc now. She figured out that these organisms just don't form colonies. They grow in these smears like this. And as long as you don't require them to form a colony, you can grow them on plates just fine. Okay, so people have counted bacteria in a bunch of different samples, and there was one study which tried to estimate how many bacterial cells there are on Earth. And the way this worked is they took different environments like ocean water and soils, and then deep subsurface soils, atmospheres, because we find bacteria in the air, and they estimated the volume of each of those different compartments, and then they calculated how many cells were present. And they came up with this number, about 5 times 10 to the 30th, which is a big number, of course. And so they tried to compare this like in Volkswagens. And one of the things they came up with was if you laid them end to end, the bacteria would stretch for 100 million light years. And they compared this to the number of stars in the known universe, which is estimated to be 10 to the 21st. That means there are about 5 billion bacterial cells on our planet for every star that there is in the known universe. More down to earth, perhaps, they estimated that the amount of carbon in the bacteria is about the same as in plants, and that there's probably 10 times as much nitrogen in these bacterial cells as there is in plants. So it's a very important reservoir for elements and nutrients on our planet, and we know that they play really pivotal roles in functioning on the planet. So I want to go back to Prochlorococcus for a second. And I meant to say this in the beginning. I realize this is kind of non-linear, and it sort of has to be non-linear because I want to talk about methods and bacteria, and I think it makes sense, but it's not straightforward. Okay, so we're talking about Prochlorococcus, and we said Prochlorococcus is a very small bacterium, and maybe 6 years ago or so, people were studying Prochlorococcus, and they realized this sort of interesting feature about it. So if you look at this scanning electron micrograph that we're looking at here, most of the cells are pretty obvious on there, but there's also all these little things, right? And those are too small to be cells. So what is too small to be a cell? So we said bacteria are small. There is a limit to how small you can be. So bacteria are membrane bound, and they have to have a little bit of DNA, and they have to have a ribosome in there. That's the thing that allows you to make proteins. So if you take a membrane and put a ribosome in the middle and then put another membrane on the other side of it, you end up with something that's maybe 100 nanometers, maybe a tenth of a micrometer. And so these things are smaller than that, and so they can't be cells. And people weren't quite sure what they were, and they started studying them. And what they realized, there's these things called membrane vesicles. They're little bits of the cell that bleb off of the cell and float away. And when they studied the makeup of the membrane vesicles, they found these really interesting things. There's DNA inside of them. Membrane vesicles themselves can sometimes fuse with other bacteria and release the contents with those other bacteria. And the function of them, or at least the ecosystem processes that they're involved in, are really diverse. So there's lots of things that eat these membrane vesicles. Remember, Prochlorococcus is a photosynthetic bacterium, so it's a primary producer, so lots of consumer bacteria will consume it. The DNA that's present inside of the cells can move to other cells, and those cells can take it up and incorporate it in their genome and start expressing the genes. So it's a way that genes move around between different bacteria, and bacteria can gain new features this way. And one of the coolest kind of things about it is that it might be a protection against viruses. So viruses recognize proteins that are on the outer membrane, and then they attach and they inject their DNA or RNA into the cell. So if you have all these membrane vesicles around, viruses will infect them, and they're not even cells, but they still have the same kind of proteins on their coat. So it might be a way that bacteria can defend themselves from viruses. Okay, so that's a couple of other points about bacteria that are really important to understand in terms of natural history of bacteria. We are used to thinking about genetics, about heredity, as being the transmission of genes from parents to offspring. So a mother gives her daughters genes, and if we look on the left-hand side of this figure, we have a bacterial cell, and it's making two daughter cells, and inside of the mother cell is that green genome, and then you get identical genomes present in each of the daughter cells. But in bacteria, there's this other process which happens all the time, and it's called horizontal gene transfer. And in horizontal gene transfer, bacteria are getting genes not from their mother, but from a cousin, or sometimes an unrelated bacterium. And they're able to take just a couple of genes and mix it in with the genome that they got from their mother. So this is a very different way of thinking about genetics than what we're used to thinking about with, plants and animals. And so this is really, really common in bacteria. And so in this figure, you can see this little red chunk here on the genome. That's a set of genes that came from a non-parent organism, which were taken up from the environment via one of a bunch of different mechanisms. And so this is an example of that. And in this example, what we're looking at are 3 different E. coli isolates. And we're looking at a genome sequence from them. And so The three different organisms are called K12. That's the E. coli that people use in the lab all the time. And then strain 536 and strain 073. And those are pathogenic E. colis. Those are E. colis that cause, the diarrhea and vomiting. And so when people sequenced the genomes from these three different isolates of the same species, what they found were that the genomes were pretty different. And this is largely because of horizontal gene transfer. So the first organism, the lab organism, had a genome which was 4.7 million base pairs long. The biggest one was about 10% bigger than that. So these are two individuals from the same species, but one of them has a genome that has 10% more genes in it than the other one. And so sort of for perspective, humans and chimpanzees are somewhere between 1% and 3% different, depending on what you consider a difference. And so here we're talking about individuals in the same species, and they're at least 10% difference. The color coding here tries to display what we're talking about. So if the organisms were all the same, then we would just see one green circle all the way around. So the green region are genes that are shared by all three. And then the colored regions are parts of the genome that were present in one or two of the organisms, but not the other one. And what you can see is a couple of things. One thing is that this is scattered all over the genome. This is not one or two events that introduce genes by horizontal gene transfer. This is something that's happening all the time. This is a really dynamic genome. And this is what we see when we look at bacterial genomes, is that individuals from the same species don't have the same genes in them. And when you look at Prochlorococcus, for example, there's about 80,000 genes that they are swapping among themselves and sharing among themselves. And for perspective, there's about 20,000 genes in humans. So even though they're these little tiny organisms, they actually have a bunch of genes that they're using for ecosystem functions. Okay, and then this is a figure, an electron micrograph showing viruses that are attacking a membrane vesicle. And so, this isn't a cell. It looks a lot like a cell, but it's a membrane vesicle. And I just thought that was a pretty fascinating process. An idea that you might have decoys out there to get rid of all the viruses. When we look at viruses in marine waters, we see huge numbers of viruses. There's more viruses than there are bacteria. And so this sort of green, starry looking image here is a sample of seawater that's been stained with this dye that causes viruses to fluoresce. And what you can see is there's as many as 100 million viral particles per mil. So viruses are far more abundant than bacteria are, and it's very obvious that viruses kill a lot of bacteria all the time. They prey on bacteria, and this is one of the main predators of bacteria. Okay, so we were talking about small bacteria, and we gave an example of Prochlorococcus, and we talked about how small the smallest bacteria are, and it has to be big enough to have a membrane-wrapped ribosome, so maybe 100 nanometers or so. There's still a lot of debate about those bacteria that are at the limits of this theoretical limit. And there's people who claim to isolate organisms that are small, and it's a pretty interesting field right now. Another very small bacterium is shown in this figure right here. And this is called Pelagibacter. And Pelagibacter is a really important marine bacterium, which is equal to Prochlorococcus in terms of mass. And it might be as much as 25% of the total biomass, particularly in deep ocean waters. So Pelagibacter is not a photosynthetic bacterium. It eats products of photosynthesis. It eats parts of other bacteria. And so we call this a heterotroph. And in particular, it's an oligotrophic heterotroph. It thrives under very, very low nutrient concentrations. And so I want to use Pelagiobacter, which might be the most abundant organism in our oceans. And it's interesting again because it was first described in 2002. So this is because we're just starting to understand some of these organisms that are otherwise difficult to study. So I want to talk about bacterial diversity a little bit. And I want to use Pelagiobacter and Prochlorococcus in order to do that. And so when we talk about metabolic diversity, so sort of at the simplest level, we often talk about what the carbon source is for an organism. For example, photosynthetic organisms use CO2 as a carbon source. Heterotrophs use organic matter as a carbon source. And then separately, we talk about what the energy source is. Is it sunlight? Is it chemicals? If it's chemicals, or is it organic matter or inorganic matter, that kind of thing. And so the two parts of that are the carbon source side of it. If we take a really reductionist view of living things, then they are primarily made of these 6 elements. And so how you get those elements matters a lot, and it describes metabolic diversity at its sort of most basic level. So we're going to look at carbon and different ways of getting carbon. And then when we talk about this, we often have 2 different varieties here of getting carbon, and this is autotrophy and heterotrophy. And we were just describing this for Prochlorococcus and Pelagibacter. Autotrophy is using CO2 as your carbon source, and heterotrophy is using organic matter as your carbon source. So this is different than an energy source. And so we are used to thinking about plants and animals, and plants carry out photosynthesis. They're getting CO2 and they're using sunlight and chlorophyll. But bacteria have all kinds of other ways of getting CO2. And animals as heterotrophs aren't using sunlight, but what we'll see is there's bacterial heterotrophs that are also using sunlight. And bacteria have this metabolic diversity because they have been around for so long. So this is a figure which is looking at the history of life on planet Earth. And the earliest life on planet Earth was probably about 4 billion years ago. And during the evolution of life, most of that time period, life was all microscopic, single cells. And so early on, bacteria evolved the ability to carry out photosynthesis, but it's a different kind of photosynthesis. It's called anoxygenic. It doesn't make oxygen. And then eventually, cyanobacteria arose, and they produce oxygen when they carry out photosynthesis. And so as a result, they added oxygen to our atmosphere. 20% of our atmosphere is now oxygen, and this is because of photosynthesis. Okay, so the first kind of autotrophy that we're going to talk about is the kind that we're used to. This is the kind that's carried out by plants and cyanobacteria, and this uses sunlight as an energy source and chlorophyll as a pigment to capture that sunlight. And in addition to having energy and CO2, you need electrons and protons, and this comes from splitting water. So water molecules are broken into their component elements, and this is why oxygenic photosynthesis produces oxygen. So it's not a coincidence that plants and cyanobacteria both use this, because when we look at chloroplasts, the organelle that plants use for photosynthesis, that organelle is derived from an ancient endosymbiotic cyanobacterium that lived inside of the plant cells and eventually just became part of the plant cells. So when we looked at those figures from Grand Prismatic Spring, these are cyanobacteria, and they're carrying out this kind of photosynthesis. And we see the different colors because they make different pigments under different temperature regimes. But so as it cools, they make slightly different pigments. And then similarly, Prochlorococcus is also using this kind of photosynthesis. There are other bacteria, though, that also are autotrophs. They're converting CO2 to biomass, and they're using sunlight, but they use different pigments for it. And there's a set of pigments that are called bacterial chlorophylls. and they differ in terms of what wavelengths of light they use and things like that. But these bacteria, instead of splitting water, split hydrogen sulfide as an electron donor, and this makes elemental sulfur. So this is anoxygenic photosynthesis. It's the earliest kind of photosynthesis, and it differs in terms of the pigments that are used and the source of the electrons. When we were looking at those purple mats, we were looking at anoxygenic autotrophic bacteria. So these purple cells here, and if you look under a microscope, this is a light micrograph, you can see that they have those little sulfur granules inside of them, just like we saw inside of Thio Margarita. And this is the byproduct of hydrogen sulfide oxidation. And they can ultimately, they can use those sulfur granules too if they have to, but they use hydrogen sulfide while it's around. Okay, then there's other photosynthetic bacteria that use CO2 as a carbon source, and they convert it to biomass, and they're using sunlight and bacteria chlorophylls, but they're not using hydrogen sulfide and sulfur. Instead, they're using things like iron and hydrogen and nitrate. And there's an organism that was isolated from Mono Lake that uses arsenic or arsenate as an electron donor. And so this is one of my favoritely named organisms, Ectothiorhodospira shaposhnikovii, which should definitely be a song about that one. And this organism uses this arsenite as an electron donor to fix CO2 in photosynthesis, but obviously this is very different than plant type photosynthesis that we're used to looking at. Then there are other bacteria, we're talking about metabolic diversity, I'm trying to give you a feel for metabolic diversity. There's other bacteria that fix CO2 into biomass, but they don't use sunlight as an energy source. And they don't use bacteria chlorophylls because they're not using sunlight, and those pigments capture sunlight energy. They directly use things like inorganic molecules as their energy source for fixing CO2. And so they're autotrophic, but they're not photosynthetic. So they're not using sunlight as an energy source, even though they are fixing CO2. And again, this is pretty different than what we're used to thinking about. So Thio Margarita is an example of an organism that grows this way, and it's oxidizing hydrogen sulfide and depositing those sulfur granules inside of the cells, and it's fixing CO2 as it's doing this. And then the iron films that we were looking at, those are growing using iron 2 as an electron donor, and when it's oxidized to iron 3, the iron 3 is insoluble, and so it precipitates out of solution. And that's what we're seeing in those troughs at Cooper Gulch and in the frog pond. Okay, so we started talking about metabolic diversity. We were talking about heterotrophs and autotrophs, and we said that autotrophs are using CO2 as a carbon source and heterotrophs are using organic matter as a carbon source, but we were differentiating the carbon source from the energy source. And that should make some more sense now. But in addition to what we talked about, there are also heterotrophs that get their carbon from organic molecules, but still use sunlight energy. And so there's lots of organisms that do this, lots of bacteria that do this. And what they're doing is they're using sunlight to drive energy requiring reactions that are different than CO2 fixation. So for example, Pelagibacter does this. And what you can see if you look at this micrograph is there's actually two membranes here, and there's this space between the two membranes. So there is a protein which is present in this inner membrane, which is based on rhodopsin. So this is the same type of protein that we use to detect light and to differentiate colors and things like that. But here, what it's using the protein for is not to detect light or color, it's actually capturing the sunlight energy and using it sort of like charging a battery with it. And that's shown here. And so this is the rhodopsin, and it uses sunlight energy and it pumps protons across a membrane, and you build up a gradient, and this is kind of like a battery that it can use as an energy source. So this is sunlight powered, but not for fixing CO2. It's just for driving general cellular reactions. Okay, so let's see. I have 3 minutes left. Um, I want to talk about Pelagibacter a little bit more. Okay, so when we first talked about Pelagibacter, we said that it was first cultured and described in 2002. And this is sort of an astonishing thing for something that makes up 25% of the biomass in deep ocean waters. So I wanted to talk about that story a little bit. So this was first recognized in 1990, and it was recognized by a guy named Steve Giovannoni, and he's up at Oregon State, and I was lucky enough to be up there at that time, and I took molecular evolution with him in 1992, and we talked about this, and he knew it existed in 1990, and he spent his next 12 years trying to get it in culture, and eventually he did, and everybody everywhere was really happy for him. So the way this came about, the way it was recognized in 1990 is as follows. When we try to identify bacteria, nowadays we often use DNA sequences to do it. And this is both because a lot of the bacteria look like each other. You don't have a lot of morphological clues, but also because it's easy to do because DNA sequencing has gotten so quick and easy. And this is a figure showing how that works. You take identical genes from different organisms and you align them. And this figure is done with frog sequences, actually. It's looking at frog evolution. But what you can see if you look at the sequences is, this branching pattern shows that these two frogs are closely related. And as you look across the DNA sequence, they have identical DNA sequences for this short region. If two organisms share a recent common ancestor, then their DNA sequences are very similar. If two organisms don't share a recent common ancestor, then their DNA sequences have diverged. So oftentimes when we isolate a new bacterium, what we do is we determine the sequence of some genes and we compare it to a database of known organisms. And the database sort of looks like this. So because everything makes proteins with ribosomes, all known living things do this, and ribosomes share features because of common ancestry, we can actually align sequences from all known living things. This is sort of an amazing thing that previous generations did not have. We didn't know how mushrooms were related to plants, were related to animals, were related to bacteria. But with these sequence comparisons, we can actually understand those kinds of relationships. And when we study it, what we see is that there's 3 groups here, and one group is the eukaryotes, and then the other 2 groups are bacteria and archaea, and these are the single-celled organisms that we've been talking about through most of our stuff. Interestingly, when you look in plant cells and animal cells, there's organelles called chloroplasts and mitochondria, and we said they were derived from bacteria that used to live inside of them. They still have DNA in them, and when we study that DNA, the chloroplast DNA groups with the cyanobacteria, and the mitochondrial DNA groups with the proteobacteria, even though these are chloroplasts from plants and mitochondria from plants and animals. Okay, so you can do this sequencing on organisms that you don't have in culture. You can take an environmental sample, like an ocean water sample or a soil sample, isolate DNA from it, sequence the genes, and then compare them to that database. And it tells you what organisms are present in your environmental sample, even if you don't know how to grow them. And so this is what Steve Giovannoni did in 1990. He was working in the Sargasso Sea, and he found these sequences which were really, really abundant. He knew there must be a ton of organisms that corresponded to those sequences that were present, but he couldn't figure out how to grow them. And it turns out that he couldn't figure out how to grow them because they live in these very nutrient-poor ocean waters. They grow really slowly, and they don't grow to a high density. So if you take drinking water, it probably has about 1 million cells per milliliter in it, but it looks perfectly clear to us. And it turns out that these cells will only grow up to about 10 to the 5th or 10 to the 6th per mil, and then they stop growing. So they just didn't know that they were growing them. But eventually, he came up with methods to grow them. Okay, so when we do these environmental DNA sequences, we find lots and lots of cases of this. This Pelagibacter is not a unique thing. And in fact, this is showing the major phyla that are known from bacteria. And each of the lines, each of the colored lines is a different phylum. The red phylum, the burgundy phyla, are, we know about them from environmental DNA sequencing, but we don't have a single bacterial isolate from those groups. So there's all kinds of bacterial diversity that we haven't been able to study because we just don't have the organisms in culture. Most of the organisms that we have in culture come from these blue phyla shown here. And the green phyla, we might have a couple, but we don't have a lot that are present in culture. Okay, one last thing, and then, the other thing that you can do with DNA sequences, because they are taxa-specific, is you can use them as stains. And so we can make probes that just bind to one kind of organism and label them with fluorescent molecules so we can see them in natural samples. And so, for example, here we see some bacillus cells and some yeast cells in a light micrograph. And if we stain them with something that stains DNA, they both stain, but we can also stain them with something that's a taxa-specific DNA stain, and then just the yeast cells light up, but the bacillus cells don't. And so we can do this. This is a sample from mud where people are using these fluorescent stains, and they're looking at several different taxa. And when you look at mud, one of the problems is there's a lot of particles in there that are the same size and shape as bacteria. So you don't know, if you're trying to do direct microscopic counting, whether you're counting clay particles or little bits of soil or something like that, or whether you're actually counting cells. And so when you use these fluorescent stains, you can actually tell that you're counting a cell and you know something about the taxonomic identity of that cell. And this has gotten fairly sophisticated, so you can now use lots of different colors for different taxa and start to understand how bacteria are organized in 3 dimensions and questions like this. Okay, I am gonna respect the time limit and stop there, but I had a quote that I wanted to close with, and I think my I think the font changed here. Okay, so this is a quote from E.O. Wilson, the evolutionary biologist, and it says, "If I could do it all over again and relive my vision in the 21st century, I would be a microbial ecologist. 10 billion bacteria live in a gram of ordinary soil, a mere pinch held between thumb and forefinger. They represent thousands of species, almost none of which are known to science. Into that world I would go with the aid of modern microscopy and molecular analysis. I would cut my way through clonal forests sprawled across grains of sand, travel in an imagined submarine through drops of water proportionately the size of lakes, and track predators and prey in order to discover new life ways and alien food webs." And obviously, as a microbial ecologist, I like that quote. So, okay, I'll stop there, and if people have questions, I'm happy to.
I have a question, Professor Wilson.
Okay, we have a question from right here.
From the peanut gallery.
Yes.
So, as a practical matter, how do the tools and methods that you use to study microbiology help gauge the health of the Arcata marsh?
Yeah, so so the question is, how do we use these tools to study the Arcata marsh? And when I was thinking about this talk, sort of my first inclination was to talk about wastewater treatment and what we're trying to do with wastewater treatment. There's 2 things that we're trying to do. We're trying to reduce pathogens and we're trying to reduce nutrients. But it's sort of hard to talk about how you do that and how you study that if you haven't talked about methods like microbiology, microscopy and genetic analysis of cells and how we think about the different metabolic diversity of cells. And so instead of trying to do too much, I thought I would sort of introduce those topics rather than specifically make it about the marsh. But we can use all of these things to study bacteria at the marsh.
So those things are like water sampling, and you would do testing.
Okay, if there are other questions, folks, you can just jump in, or we have a hand signal. Where is that?
There's a little place you can raise your hand,
Oh,
a little blue
There's one
hand.
from Ann Kirby. Go ahead.
Thank you, Ann Kilby. You said that you could have two individuals of the same bacterial species, but different DNA in them. In us macroorganisms, we're used to saying that what defines a species is the genome. Do they reproduce together or not? And so I'm wondering what actually a species is in that context.
This is a great question, and mostly we have operating definitions of what a species is, and we don't necessarily, some people do, but many people, including me, don't believe that the species concept really applies to bacteria. So the biological species concept is based on the ability to reproduce together, and these are asexual, and so they don't interbreed, and so the biological species concept probably doesn't apply. But the reason why the biological species concept makes sense is that if organisms are interbreeding and sharing genes, then they tend to be more similar to each other genetically and different from things that are other species. So there's some genetic cohesiveness to it. And there might be some kind of genetic cohesiveness to bacteria, and it might have to do with horizontal gene transfer. So these organisms that are capable of moving genes around, they still tend to sort of converge in genetic units, and that applies to a species. But it also might be that what happens changes over time. So you get species and the individuals are diverging and diverging and diverging, and then there's some selection event and it all collapses back into one sort of genetic unit. So where you are in time is going to help you define what a species is.
Neat, thank
But that's
you.
a great.
Okay, thank you, Ann. I'll keep looking for hands. I wanted to thank you for that quote from E.O. Wilson. And am I remembering that he's such a good writer that he's won 2 Pulitzer Prizes?
Mm-hmm.
He wrote a bunch of popular science books which were really accessible. And he also did great research in sociobiology and ant biology. Yeah.
Ants, yeah. All right, while we're waiting for another question, I have one. We're used to seeing how humans have disrupted biology at the macro level, and I'm wondering if our pollutants have disrupted the environment that you are in touch with. Dioxins are all over the marsh, for example.
Right, right. This certainly happens on lots of scales, and typically what ends up happening is human activities end up selecting for one group of bacteria over another. And so from our perspective, this causes problems. From the bacterial perspective, probably not so much. But for example, we were talking about wastewater treatment. So one of the things that you're doing with wastewater treatment is you're trying to get rid of the nutrients. And one of the reasons why you're trying to get rid of the nutrients is because the water has to go somewhere. So when nutrient-rich water goes into another body of water, then you often get things like algal blooms. You get lots of growth. And then bacteria will break down the algae. And as they do that, they use up the oxygen. And so whatever the receiving water is will go anoxic. And this kills off fish, and it kills off invertebrates, and it can kill off all kinds of things. And this happens now all over the world. Where the Mississippi River enters into the Gulf, there are anoxic zones, and it's caused by nutrient runoff from farms. In the Chesapeake Bay, there's big anoxic zones and it kills off all the crabs and all the fish. And so there's lots of human activities like that that disrupt ecosystems. And because bacteria are central to ecosystems, they play a role in these things. There's also lots of bacteria that break down pollutants that have that ecosystem function. There's bacteria that can grow using plastics as a carbon and energy source. And so they go slowly, they break them down slowly, but they can use that as a nutrient source.
Huh. I think there was a European group that came up with enzymes. I wonder if they were from bacteria that can eat plastics. And so maybe there's a way out.
Yeah, no, the enzymes came from bacteria. Yeah. The way I first found out about this story actually was, as a faculty member, we got approached by local elementary school students to help with science fair projects. And I had a young woman approach me, and she wanted to study plastic eating bacteria. And what we ended up doing was a sort of a bioinformatics study, a computer gene-based study, which she ended up doing. But I taught her how to do it. And so what she did was she looked for those enzymes that break down plastics. And what she found, there's two main ones. She found that one of them was present in a bunch of organisms, and the other one was present in a bunch of other organisms. And so her idea was, in order to break them down faster, you have to get some kind of culture where you're growing both of those organisms at the same time. And then they can sort of cooperatively work to break the plastics down, which I thought was pretty neat for somebody who's in elementary school to come up with.
Yeah,
Yeah.
young PhD student working there. Okay, are there other questions out there? Feel free to unmute yourself and jump in.
I'll ask a question. So extremophiles, I think, are usually, or at least I think of them as being in very hot environments, like volcanic areas. Are they in very cold areas? And do people study that? And I'm thinking more as in like Mars, the water that might be on Mars. Could that ancient bacteria that is there, if there is there, could that be something that after hundreds of millions of years or billions of years still be something we could resurrect?
It probably doesn't even need to be resurrected. My guess is that there's life all around the universe. There's all kinds of things that are living. And so extremophiles, that's a very anthropomorphic term because it's extreme to us, but they like it, right? So an organism that grows in boiling acid would find pH 7 and 20 degrees C to be absolutely horrible, horribly extreme, way too cold, way too difficult a pH to live in. But extremophiles are fascinating, and when we look at the extremes of things, we see organisms that, for example, can grow at 122 degrees C. That's about 250 degrees Fahrenheit, which is way above the boiling temperature of water. But these are organisms that grow at things like ocean depths and hot vents in the ocean. But they can grow at temperatures far above the boiling temperature of water. Then there's other bacteria that can grow down to minus 20 degrees C, so well below the freezing point. And they're typically growing in salt-rich waters so that the water is super cooled, but not actually ice. And there's bacteria that can grow at -1 pH. There's bacteria that can grow under high pressures, high amounts. There's bacteria that can grow in nuclear reactors. They're so good at evading radiation. So extremophiles are fascinating, and there's a very active research program. The field's called astrobiology, and astrobiologists are trying to understand what kinds of studies you would do to detect life on other planets. And there are some things we can do from here on Earth to find life on other planets. For example, we talked about the idea that because we have photosynthesis, oxygen concentrations have built up to 20% of our atmosphere. Oxygen is very, very reactive. And the only reason that we're at 20% oxygen is because we have all kinds of life forms pumping that oxygen out. So we could look for very reactive molecules in the atmosphere of other planets. And if we find it, then that's a good sign that maybe there's life on that planet that's creating that reactive molecule.
Thank you.
Like that molecule they were looking at on Venus, in Venus's atmosphere? I forget what it was called.
I actually don't know about that.
Yeah, well, a follow-up story a couple weeks later, this is recent, said that it might be a mistake, but somebody spotted something in the atmosphere of Venus that was a potential life source, and it might be from something like you were just describing. Okay. Other questions, folks? Well, we promised to expose you to the secret life of the marsh, and we ended up traveling the world with the microbial universe this evening. So a round of applause and thank you to Mark Wilson for the time and information. And one last thing, we will publish the recording for this lecture and make it available to anybody who wants to go over some of those finer points that you just saw. So it will be in our newsletter, and our guru for that sort of thing is Sue Leskew, and she'll have it out to membership and so on. So thank you all for attending. Thank you, Mark Wilson, for a really interesting evening.
Thank you, Mark.
Thank you all. Thank you all very much. I'm glad that you wanted to hear about bacteria, so I appreciate that.
Well done.
Bravo. Thank you.
Thanks.
Thank you.
Okay, good night all.
Goodbye.
Bye.